you for joining the virtual GlyGen and the Common Fund Data Ecosystem Bioinformatics 2024 workshop. So this workshop, the three of us, Subarna, Jeet, and myself, we presented it at the Bioinformatics 2024 meeting and conference in Delhi, India. And this is the same workshop in a virtual format because there were several people who could not attend the meeting because we were full. So hopefully there are people who could not attend that day could join today's meeting, and maybe a few others who are able to join. This meeting is being recorded and will be shared publicly. So if there are people who could not join today, they want to take a look at it, they can review the slides and the video later on. So this is a work, there will be a combination of slides that I'll show, and then also there will be some hands-on activities that people will participate in. So first thing is that please, I'm going to wait a little bit. I want you to take a screenshot if you can of this, because this URL I want you to save. So go to your browser. So https wiki.glygen.org, GlyGen _ there's an underscore here, CFDE _ Bioinformatics 2024 _ workshop. So this is the page that we will be going to when during the hands-on section of the workshop. So if you can, please type it in and bookmark this page. All right. So my part of the talk is the first 20 minutes, and I'll be talking about protein bioinformatic analysis and few housekeeping things. I already mentioned recording. Please stay muted, turn off camera, use the chat for questions, unmute yourself for Q&A when we do the Q&A session of this workshop. So first I will be presenting, and then second Jeet is going to come in, and last is Subarna, and finally we will have Q&A. So this is a 2-hour workshop, and we plan to continue. The first hour would be more presentations, but the second part of the workshop is use case discussion, and also we will try out a few use cases, time permitting. So where are we? At least Jeet and myself. So both of us are George Washington University on the left side, and this is the metro station, Foggy Bottom in Washington, D.C. It's pretty close to the White House, almost walking distance, and our office is right outside this metro. This is the entrance. We are very close to Kennedy Center, which is a really nice place for arts and theater and music. Again, like 7 or 8 minutes walking distance from our office. So it's a really nice place. For those of you who haven't been to Washington, D.C. at some point, do try to come and visit us. So who the GlyGen team, it's a big team, but not everybody is working 100% on GlyGen. Everybody or most people are working on more than one project, but this is the team on the left, and Mike and I are the principal investigators, and we have our advisors who advise us on GlyGen, and the project is funded by 2 NIH awards. One is a R24 award and one is a OT award. So it allows us, and I want to make sure that I acknowledge them, it allows us to do what we do and also improve upon the things that we get feedback from. Like today, hopefully we'll get some feedback and we can use them to improve GlyGen and also Common Fund Data Ecosystem. So in an earlier slide, I had mentioned to use this link. This link is again here, https wiki.glygen, GlyGen _ CFDE, and so on. So on your cell phone or your device, computer or whatever have you, if you are on that link, and I have been taking screenshots in these slides, you will see a lot of screenshots of a cell phone interface. And the reason I'm doing this is because I feel that many of these workshops, it's very hard for everybody to come with a laptop. All of us, I would guess, has a cell phone with internet access. It's much easier to do these workshops using a mobile device. So this is in Delhi, we did it the first time. We used this format, and it was quite successful. People could follow on using their cell phone. So if you go there, you will see, scroll down, and you will see a link called important links. And if you click on that, you will get to a Google Doc page, but I'll come to that later on. So this is where there'll be other links in there. So a little bit of history regarding why am I using, or why are we using, glycans and glycobiology and glycoinformatics as a starting point. So in 2012, I mean, everybody knows how important proteins and genes, and you may have heard a lot of talks on them. So I wanted to show that, proteins and genes can tell part of the story, but glycans can tell another part. So when we are talking about bioinformatics, integrated bioinformatics analysis, we need to understand that, just genes and proteins, they are not on their own. There are other things that involve the other biomolecules that are involved in the final outcome of any kind of environment or disease or whatever have you. So in 2012, there was a great, this roadmap for the future was published, Transforming Glycosciences. A little bit of history. Before that, a lot of work on research, of course, had gone in. There are lots and lots of papers before that. But 2012, it really started coming into this glycoinformatics space, the way at least I see it. And then 2014, the Glycosciences project started by NIH. And then in 2015, there was a funding opportunity that we applied to, and we were successful. It was a planning grant on how to organize glycan and proteins and related data. 2017, William York and myself, we got funded to do the work. Later on, Dr. York retired, and Dr. Tiemeyer is now a co-PI with myself in the current project. So again, why glycans? This is a really nice figure. It was by Dr. Morman and others. Vertebrate protein glycosylation. It shows about the diversity. So you have the proteins, and you have all kinds of glycans attached to it. And these glycans, and we all know about the SARS-CoV-2, right? When we look at the glycoprotein and the picture of SARS-CoV-2 that you most probably are familiar with, the spike protein, you will see all the glycans that cover. And glycans are really important, not just for viruses, but for a variety of other organisms, and it can affect the final outcome. So glycoconjugates, whether secreted from cells or found on the surface or inside cells, they regulate functions. They are almost all currently marketed biologic pharmaceuticals are glycoproteins, antibodies, growth factors, enzyme replacements. Glycan structural features influence glycoconjugate function. So the question is, where is this data captured? So most of the time when people do bioinformatics, as I said, you are working at the gene level or genome level and then the protein level. But this type of information can actually start thinking at a systems level approach to bioinformatics. Now, what is most confusing for people when they start getting into bioinformatics is there are thousands and thousands of resources, and who has the time and even the knowledge to go where to go to find information. So from a database and a knowledge-based perspective, I think it is our role as developers of these resources at an international level, not just in the US level or Europe or Asia, India, whatever place you are at. We need to work collaboratively at an international level so that we are sharing data, talking to each other, and also producing resources which are interconnected with each other. Otherwise, if everything is siloed, then people will not be able to find the information they need. So we started this GlySpace Alliance with collaborators in Japan and in Europe, in Switzerland, Swiss Institute of Bioinformatics. And we share data. And one of the biggest things that we did is that when we are sharing data, we use a license, a very clear license. And anybody who is planning on developing resources, think about these licenses when you put data out. In our case, our licenses, anybody in the world can use it, including commercial entities. We don't care. We actually want people to use it as much as possible. The only thing we recommend or suggest is that they cite our publication when you use these resources, because end of the day, these citations also help us understand how people are using the products that we make. Today, we will be using 2 proteins as an example. So this is you don't have to do this right now, but we will come to it. So this is one protein, and this is the second one. And there are a couple of papers I have on these slides, which you can explore later on. So some basic background. This is a great figure. I like it, the tree of life. In 1969, Whitaker summarized the 5 kingdoms. So, how our understanding of the tree of life has changed and is still changing. And then in 1990 is where we stand right now. You have eukaryotes, you have bacteria and archaea. These are the 3 major kingdoms of life. And when you think about it, a lot of the work has been done in the bioinformatics space to better understand the tree of life and understand the basic cellular function. And I cannot not talk about Dr. Dehoff without, she started a lot of these things. She's a bioinformatics pioneer. She was a physical chemist, and she was interested in possibility of deducing the evolutionary connection in the biological world. And this was the first database, but it's in the form of a book, but this was the first database that she published of protein sequence database. And this figure many of you may have seen. This figure shows, if you have used BLAST to look at sequence homology, this is what it is. So she did alignments, and others later also did it, and tried to understand which amino acid can replace the other one. And you put a score, and you can use these scores to identify homologous sequences. And that has also revolutionized science. If you know, BLAST, the publication BLAST, which many of you may have used, is one of the highest cited paper in the world. And it tells you, where bioinformatics is and how important it is. This is the NCBI website for BLAST. I'm not going to go into that other than show you an example. In a BLAST result, the red ones are very closely related. And then when you see green and this pink color, it possibly are different domains within the protein. And using BLAST, you can actually get a lot of information when you are trying to understand protein function. And using BLAST, you can also tell if I have a protein which has homologs in plant, fly, human, worm, and yeast, most probably it's 1.7 billion years old, and it's really important. So you can actually start using these BLAST-like tools to identify which proteins are critical for a particular organism. And actually, we're also present in the last common ancestor 3.5 billion years ago. So in terms of annotation, what is annotation? In the Merriam-Webster dictionary talks about it as a note or added by way of comment or explanation. And I want to go over a few concepts of how to do protein annotation. So what is known? If you have a paper that describes a protein or the glycan and had done experimental characterization, that is known. What is obvious? When you have homology and you can say these proteins are very closely related, then you can say most probably the function can be transferred. But the caveat is that if the glycosylation sites are not conserved, then the glycosylation may not happen. So there are non-obvious ones when they're distantly related, and these are known where they're protein, they might be conserved, but none of the proteins within that family have any papers associated with it, which talks about characterizing that protein or gene. So in those cases, you know that this protein is conserved, but function is not known. So I'm going to switch gears a little bit to give a couple of things that, examples. So this was a paper written by Dr. Rolf Achweiler. Right now he's the joint director of MBI in Europe. And he wrote this paper with others in 1999 and talked about glycosylation, 75,000 entries, 2/3 of which include N-glycosylation's consensus sequence. So they concluded that the majority of this type of site, asparagine, some other amino acid, serine or threonine, is glycosylated. So you look at I started, looking at other papers. This is connecting this to diseases. So there is this paper, and this text is from that paper. 3 children have the same interferon gamma receptor missense mutation, have a particular disease. So they get infected very easily with certain bacteria. So there's a mutation present in a patient, which makes them susceptible to a particular disease. And that's also at the crux of a lot of the genetic testing and the future of what genomics and proteomics holds, right, in terms of health and disease. So when I look at this paper, I found this another paper in 2005. So this is how, you know, bioinformatics research usually starts off. You start looking at things and trying to understand what's going on. So there was a part bioinformatics research paper. It came out in Nature Genetics. And this table showed that due to missense mutations that are changing the amino acids, there is gain of glycosylation. The asparagine or the serine or threonine is gained, and a glycan can be attached in that position, or there's a loss of an existing site. So when you look at this type of data, you say, okay, that was a prediction in the bioinformatics space. And sometimes it takes a lot of time for people in the wet lab to prove or disprove some of those predictions. There was a paper in Nature 2019, which showed that that type of gain of glycosylation or loss of glycosylation actually can affect the protein structure, and it results in steric clashes. In this case, the addition or gain of glycan structure or glycosylation structure results in clashes, which there was likely to prevent the interferon gamma receptor 2 from docking to the high affinity intermediate protein complex. So this is how one would, but how did I find this? Because of the knowledge base. So when I was preparing for this talk, when I was trying to, you know, do this workshop earlier on, I went to GlyGen and I went to this protein, and I knew about the 2005 paper. And then in the same entry, I found this paper, and that's how I came to find this paper, that, okay, in that same position, there is actually another paper. So as a source of information, yes, Google, you can Google things, but sometimes you don't even know the prompt to Google. Same thing, you can do chat. By the way, I tried this in ChatGPT and it could not find the answer. So yes, there's large language models which are going to be great at some point and is already great in some respects, but there is still no easy shortcut ways to get to information unless it is organized in knowledge bases. So here is an example of how the entry looks like. In that particular asparagine, a glycan is attached. You can click on the link later on and see how the glycan gets attached. On the left, it shows you how you can also highlight the different sugars, the mannoses and the galactoses in this entry. Again, part of this talk also is we might not be, because it's going pretty quickly, you might not be able to click and do what I'm saying, but in the second half of today's, the second hour, we will have some opportunities for you to try some of these things out. And this is a very high level view of how the data is organized, kind of a data model. So we have proteins, and then within the protein, there is a site, and within that site, a glycan can be attached. Proteins can be annotated with a bunch of different information, biomarker, sequence, variation, phosphorylation, disease, and so on. Same way, glycan can also be annotated with this. So having a good data model to organize your data is also really key. And later on, Suvi is going to talk about some of the way this data is organized in the common fund system. So these are the organisms in GlyGen. We don't have everything. We go organism by organism because it's important that we have really high quality data. And I'm back again to that important link. By now, you might have gone to this page. Again, on the right-hand side, there are some links you will be using later on today. The easiest way to get to a protein, if you know the gene name, I sometimes just go to Google sometimes to do GlyGen space EGFR is the gene name, and you get the protein. Or, of course, you can go to glygen.org and search for that EGFR. Just type in EGFR and you will find it. You don't need to do this right now, but I'm just giving you some screenshots. Once you get to that EGFR protein, you can scroll down, you can click on glycoproteins, you can click on other things in there, or select boxes in there to get more information or filter the data. This will also be shown later on by us. So you don't need to right now. So you can click on the accession number and you can go to glycosylation, and on glycosylation, you can click, again, this will be more later during Jeet's demo, you can scroll down and see some of these things. Finally, you can also download what you see in form of a table, Excel file or Google Sheets. And you can also go from, and one of the important things when you are working in bioinformatics is that sometimes you want to go from resource A to resource B. In this case, if you want, and they're all connected through close collaboration with other resources. So in this case, EGFR human, the UniProt entry has additional information, right? So you can click on it and go to the UniProt entry. UniProt is another project which is not run by us, right? It is run by groups at Georgetown University, European Bioinformatics Institute, and Swiss Institute of Bioinformatics, and is largely funded by NIH also. So if you go to UniProt, from UniProt, they also connect to NCBI, which is again a completely different group at NIH. NCBI is in Bethesda, maybe an hour or maybe not half an hour from our offices. So if you click on RefSeq, scroll down, and the same protein you can view on the right-hand column as a RefSeq entry. So RefSeq protein entry. Common Fund, Suvi is going to talk about. Lots of projects also is trying to integrate data. Sometimes you have to go to a place like Common Fund to get data at a more granular level. And you can also look at knowledge graphs and a variety of other things in the Common Fund data ecosystem, which is also an NIH-funded project. Several PIs are involved. With that, I'm going to stop. Please feel free. So if you go to the GlyGen website, you can click on help, and then there is a place where you can get contact, and you can send your question, message, things that you might want to do. If you want to have a demo for your lab, or you know, let us know. With that, I will say thank you and for listening, and I'm going to pass on the slide or screen sharing to G.